All right, so I think everyone is back after the tea break. So we'll continue our sessions, and this will be young investigators' presentations. I would like to welcome Professor Jyoti Tamang and Dr. Sharad Gopalan onto the dais. So both of them will be chairing for young investigator presentations.
Did we start?
Hello. No, no. Immediately. Those who are outside, please.
Yeah, they should be here.
Ladies and gentlemen, for your kind attention, if any of our young investigators are outside the venue, outside the hall, please come inside and join for the session, because this is something really important, and we'll definitely have some thought-provoking questions or session. So requesting all our young investigators to join together inside the hall.
Start? Yes, start. Good afternoon, dear colleagues. This session is actually a very, very important session, because if you actually look at the future of documentary evidence when it comes to scientific literature and any new work going on in the field to update hard evidence-based information, it all depends on active research. And the future of these active research initiatives are to be taken by a younger generation. It is our youngsters who will lead the way. And this Young Investigator Award is a very, very important session from that standpoint, because these are the youngsters who have done original pioneering work, which may have a huge applicability and may influence not only certain clinical decisions with applicability, but also in some situations, maybe policy changes. So this becomes an important session, and we really are happy that we have had a number of young investigators coming forward to present their abstracts in this symposium. And they were really high quality abstracts. I now request Dr. Jyoti Tamang to tell us more about the Young Investigator Awards, how these awards are processed, how one went about it. Please go ahead, Doctor.
Namaskar. One of the very important flagship program of our uh, foundation is to encourage the young researchers or young investigators working in the probiotics and the related fields. And every year, along with our international symposium, we also organize the poster awards, posters for the young investigators. So the process is that we invite the posters in our website and our contact to all the university institutes by our secretariat, mostly by Dr. Nirza. Then after that, we get a nominations, we get a applications. Then they do the screening. After that, they evaluate the applications. The poster, the abstracts are being evaluated by the members of the governing body members of our foundations. But the condition is that the students of the member will not be evaluated by, suppose if I have a students, I cannot evaluate my own students. So that is the thing. Then after that, we select the best investigators, and they are asked to present here, come, we'll provide their travel and the other logistics. Then they are asked to display the posters, and there is a jury. The jury is not from our GUT Foundation members, as an international independent jury. So they select first, second, third every year. So this year we have received 51 applications. Out of 51 applications, 20 have been shortlisted, they have been selected. You are here, all 20. You made your presentations, you made your the poster display, and our international jury has already evaluated, and the marks is with me. So with my friend Dr. Gopalan and me, we have the list. I will go with the third list, third name, third prize, then second, and the first. So when I announce the name, you can just stay there. Then after that, we will invite one by one for your presentations, only 10 minutes presentation, exactly 10 minutes presentation, and there will be no question answer, and you will be awarded. The third prize goes to Aditi Goel of CFTRI, myself. Please stand up. Where is Aditi? Okay. Second prize goes to Akashi Gaike of SPP University. Now we have first prize, but there are two equal marks. So I'll go by the alphabetic name. First, Ayushi Purohit of the Translational Health Science and Technology Institute. Then first, another first is Manusurya Palapu. Okay, now I invite Ayushi Purohit to make a presentation, 10 minutes exactly. And you'll be given the warning at 8 minutes. No, no.
So good afternoon, one and all. So my abstract was entitled as a gut signature linked with increased ethanol production and liver inflammation contribute to non-alcoholic fatty liver disease in Indian patients. Yesterday, I had a talk about this topic, but today I'll go in more crisp details. So non-alcoholic fatty liver disease terms refers to a range of spectrum of disease with excess accumulation of fat in the liver. If the fat in the liver is more than 5%, it is known as non-alcoholic fatty liver. The spectrum of pathology start with steatosis through inflammation to fibrosis and ultimately leading to hepatocellular carcinoma. This silent epidemic is on horizons because it has no therapeutic to the disease. So there is a bidirectional link between the microbiome and the liver that is known as the gut-liver axis. The portal vein carries all the microbes and the microbial metabolites and transmits to liver, which is one of the critical risk factor for the disease. This disease is on horizons because there is no therapeutic till date for NEFL. You can just reverse by consuming 800 units of vitamin E, weight loss diet, regular aerobic exercise, eliminate alcohol. If diabetic, tight control of glucose level, lose 7 to 10% of the total body weight. So the key risk factors that associates from transferring to NEFL to NASH, which is one of the severe form of the disease, are genetics, bile acid, gut dysbiosis, lipotoxicity, and environmental factors. So I have worked concurrently on gut dysbiosis part. So the sampling was done in the NCR region of Delhi. We collected the sample from 3 cohorts: healthy, obese, and NASH. Healthy were 22, obese were 17, and biopsy proven NASH were 10 that were done with Dr. Shalimar from AIMS. So this is the characteristic of the people. We took the diet that is the vegetarian, vegetarian, and the non-veg diet, and we took the biopsy approvals from the AIMS. So when we calculated the core OTUs, we found 12 OTUs were present in all 3 cohorts, that is healthy, obese, and NASH. Based on the median value, we calculated and we found Colensella aerofaciens was one of the taxa that was increased rapidly from healthy to NASH. As obesity is directly linked with NEFL, we tried to illustrate the genomic repertoires of Colensella aerofaciens in Indian population. We took biopsy-proven NASH, isolated this Colensella, screened it with the specific primers, and did the whole genome sequencing at THSTI at MySeq platform. The complete genome had 2.3 MB in length, 60.1 GC content. The genome had enriched itself in the virulent 29 to 30 more genes that were virulent, and more than 258 genes were involved in carbohydrate, fatty acid, and lipid biosynthesis. So there was a remarkable feature from our study that we found a SNP that is present in the NASH isolates that was mutated and was leading to high alcohol production in the gut. So if these people are the people that were not having alcohol, but the bacteria was making alcohol in the gut. So this is the amino acid sequence that we have sequenced. You can see the mutation. This reference is from the healthy person, and this is the mutated gene at the SNP level of all the isolates that were isolated from the NASH individual. We started with the animal study. We took C57BL6J male mice, 8 to 12 weeks old. This was the schematic of the work that we have done. We have taken the normal chow diet and the CDHF. We have given 2 doses of the bacteria subsequently after the weeks at day 28 and day 35 and given through the oral gavage. We took the body weight, the serum samples, and the liver for the downstream processing. So these are the things that we have measured. We have measured the circulatory ethanol levels, the fibrotic parameters, inflammatory parameters, lipogenesis and lipolytic parameters, hydroproxylene, triglyceride, and histopathology analysis was done. So this is the triglyceride analysis. You can see the mice that were fed with CDHF and Colensella showed a high increase in the triglyceride level. The macrosteosis you can see in the cohort of CDHF plus Colensella showed macrosteosis and microsteosis. It means that is what inducing the inflammation in the liver. This is done by GCMS. We took the mice blood and calculated the ethanol concentration. So you can see the mice that was fed with normal chow also, but if given Colensella aerofaciens was increasing the ethanol production in the mice liver. So the hydroproxylin and the triglyceride ratios were also increasing with CDHF plus Colensella aerofaciens. These were all the inflammatory markers and the fibrotic markers like NF-kappa beta, TNF-alpha, IL-6. IL-10 is an anti-inflammatory marker. You can see in the cohort with CDHF plus Colensella, it was tremendously decreasing. Alpha SMA, Col1, alpha TGF beta are fibrotic markers. We also seen the fat uptake and the fat releasing genes like PPR alpha and FAT-P5. So you can see that FAT-P5 and PPR gamma was increasing with Colensella aerofaciens. So the conclusion of the study is we study the gut microbiome of NEFL, obese, and healthy subjects. We observed a higher abundance of Colensella aerofaciens in the obese and NASH patients. Cholinceriofusion contributes to increase in the level of systemic ethanol in the mice. Increased ethanol, hydroproxylene, and triglyceride induce hepatic inflammation. The insight of this current finding is the first study to correlate the genomic signatures of cholinceriofusions and association with NAFLD and disease progression. So there are several limitations of the study. In this study, host bacterial interactions were not explored. The study are primarily focused on the bacterial taxa, as we know that microbiome is coming in limelight, which based on the fungal component has not been elucidated. This has been done only on 10 samples, so it is having a limited sample size. The cross-kingdom interactions have also not been done, so this study has to be validated in a larger cohort. This is the graphical abstract. We have seen that low level of Collinsella aerophaciens lead to low ethanol production, which can lead to less inflammation, but high abundance of Collinsella can lead to high ethanol and high inflammation. We have done the NGS, GCMS, qPCR, and microtome. So if you want to go in detail, you can go through the article that has been recently published, Collinsella aerophaciens linked with increased ethanol production and liver inflammation contribute to pathophysiology NAFLD in eye science. So the minds between this study are Dr. Bhavatosh from THSTI and Dr. Sanjay Banerjee from NIPER Guwahati, who are my supervisor in this study. So thank you.
Thank you, Dr. Ayushi.
Thank you.
Thank you, Dr. Thank you, Dr. Ayushi. We now move to the second speaker, who's also got the first prize. Manisuria Palepu from the National Institute of Pharmaceutical Education and Research, NIPER. Over to you.
Good afternoon, everyone. Good afternoon, everyone. My name is Mansurya Kumar Palepu. I am from NIPER, Hyderabad, and I'm working under Dr. Manoj P. Dandekar. This is my thesis work. The title is Short-chain fatty acid supplementation rescues anxiety and depression-like phenotypes generated by fecal engraftment of treatment-resistant depression rats. Treatment-resistant depression. This is the basic pathophysiology of the depression. In this, there is an alteration in the HPA axis and the tryptophan metabolism, and there is an increase in the neuronal inflammation and neuronal death, and the neurotransmitters get dysregulated. Rational. In the major depressive disorders, we have given this, like when we give the antidepressants, some are responders or some are non-responders. So here we are elucidating how the gut microbiome composition differs between the responders and the non-responders. That may be the novel therapeutic treatment. This is the methodology. So rats were subjected to the chronic unpredictable mild stress for the 6 weeks period of time, and adrenocorticotropic hormone for the 2 weeks period of time. Yeah, 2 week period of time, and we have given the short-chain fatty acid treatment for a 1 week period of time, and we did the fecal microbiota transplantation from the control rats to the healthy recipient rats, which was named as control FMT rats. And we did the fecal microbiota transplantation to the from the TRD to the healthy recipient rats, which are the TRD FMT rats. These are the methods and instruments we have used in this study. Coming to the results and discussion. Here you can see we performed the behavioral parameters, sucrose preference test, forced swim test, and open field test. And in these, the depressive like behaviors were increased in the TRD and the TRD FMT rats significantly. And after the administration of short-chain fatty acids for a 1 week period of time, it got reduced. And coming to the anxiety-like behavior, we did elevator plus maze test. And here also you can see the percentage entries in the open arm and percentage time spent in the open arm are decreased in the TRD and the TRD FMT rats. And it got reversed when we given the short-chain fatty acid treatment for a 1 week period of time. And coming to the FFAR receptors, free fatty acid receptors, we did the RT-PCR for the FFER2 and FFER3 receptors in the frontal cortex. And you can see there is an increase in expression of the FFER2 and FFER3 in the after giving the short chain fatty acids as a treatment. And we quantified the corticosteroid and the BDNF. In the plasma, the corticosteroid levels are increased in the TRD and the TRD FMT rats, and after giving the short chain fatty acids, the levels got reduced. Next, we quantified the BDNF levels in the frontal cortex. You can see in the TRD, the levels got reduced, and after giving the short chain fatty acids, the levels are increased, restored. We also did ELISA for the pro-inflammatory markers for the TNF alpha and IL-6 in the frontal cortex and the hippocampus. So you can see the TNF alpha levels are increased in the TRD rats and TRD FMT rats, and in the frontal cortex we have not observed any significant different changes in the TNF alpha levels, but we observed significant changes in the TRD FMT rats. And coming to the interleukin-6, in the normal group, we have not observed any changes. But in the FMT groups, after giving the short chain fatty acids, the level got reduced. And the interleukin-6 in the hippocampus, there is no significant changes in across the groups. And we did RT-PCR for the C-reactive protein. The C-reactive protein increased in the TRD, and after giving the short chain fatty acid treatment, it got reduced. And we also estimated the BDNF and double coating levels, which are neurogenic markers, and the levels got reduced in the TRD and TRD FMT rats. Octreotide, we also did the mRNA expression of the octreotide. This in the octreotide levels got reduced in the TRD and the TRD FMT rats, but we have not observed any changes in the FMT groups. Coming to the neurotransmitters, we quantified the neurotransmitters in the LCMS triple quadrupole. You can see the levels of serotonin, dopamine, GABA, and norepinephrine are reduced in the TRD and TRD FMT rats, and after giving the short chain fatty acids, the levels were restored. And the SCFS reversed the altered tryptophan metabolites. The tryptophan and the cyanuric acid are they are having the protective nature. These levels got reduced in the TRD and the TRD FMT rats. And after the short chain fatty acid treatment, it got reversed. And the cyanuric and the quinolic acid are the neurotoxic compounds in the brain. These levels are increasing in the TRD and TRD FMT rats. We performed the immunohistochemistry of corticotropin releasing factor in neurons in the paraventricular nucleus region of the hypothalamus. In the TRD and the TRD FMT rats, you can see the CRF neurons, but after giving the short chain fatty acids, in the TRD1, it got not reversed, but in the TRD FMT rats, it got reversed significantly. And we performed the immunohistochemistry for the jonocodin protein in colon, and you can see the B and E, TRD and TRD FMT rats, the expression levels of jonocodin got reduced, and after giving the short chain fatty acids, the levels are increased significantly. And we performed the short chain fatty acid concentration analysis through the NMR in the plasma and fecal samples. And you can see the levels of acetate, propionate, and butyrate got reduced in the TRD. There is no significant changes in the levels of acetate in the plasma, but there is significant changes in the levels of propionate and butyrate after giving the ACFS. And we performed the ACFS concentration in the fecal samples also. And here you can see there's a reduction in the acetate, propionate, butyrate in the TRD and TRD FMT rats. We did a we did a HNE staining and Elsham blue staining. In the TRD and the TRD FMT rats, the Villi to crypt ratio got reduced, and you can see the neutrophil infiltration in the TRD. And after doing the short chain fatty acids, it got reversed. And in the Elsham blue staining, the number of goblet cells are reduced in the TRD and TRD FMT rats. And after the treatment with HCFS, the number of goblet cells are increased. And on top of that, we performed the 16S metagenomics. Here you can see the some different changes are there in the relative abundance in the phylum level and the family level. And here we observed changes in the genus of Faecalibacterium, Subdoligranulum, Solobacterium, and Peptococcus in the TRD as compared to the control rats. A conclusion is rats which are exposed to chronic unpredictable mild stress and ACT has showed the depression and anxiety-like behavior. And rats induces the changes in the gut microbiota composition. The short chain fatty acids reduce the levels of TNF alpha and IL-6 in the frontal cortex and hippocampus. And there is an increase in the CRF expression in the paraventricular nucleus of the region of the brain. And the short chain fatty acids normalizes the levels of neurotransmitters like serotonin, GABA, norepinephrine, dopamine in the frontal cortex, and tryptophan metabolites in the plasma. And it got the levels of neurogenic markers like BDNF and W-gluten are reduced in the hippocampus. The keynotes. Short chain fatty acids, they may have the beneficial effects by the modulation of tryptophan metabolism, inflammation, neurotransmitters, and neurotrophic factors, and the brain access through that. So robust clinical trials are required to validate the potential therapeutic effects of the short chain fatty acids in the TRD. We are thankful to the NIPER Hyderabad Department of Pharmaceuticals, Ministry of Chemicals and Fertilizers, and IBRO International Brain Research Organization for their support in conducting the present study. And this work got accepted, in 2 weeks back in the ESS Chemical Neuroscience, and that one is the cover page in the ESS Chemical Neuroscience. It also got accepted. Thank you.
Congratulations, Manasurya, that you got this accepted. It's a good impact factor. We now move to the next presentation for the second prize, Akshay Gaike from the National Center for Cell Science and Department of Zoology, SPPU. Akshay, please come.
Good afternoon, everyone. Myself, Dr. Akshay Gaike. I am the title of my abstract is title of my study is effect of long-term oral glutathione supplementation on gut microbiome of type 2 diabetic individuals. I work with Dr. Yogesh Sauche from NCCS and Professor Saroj Gaskadbi from SPPU Pune. So diabetes is a complex multi-system disorder and its prevalence is increasing worldwide. It's around 2 to 20% of the global population is affected due to diabetes disease. In India, it constitutes around 19.3% of total number of diabetes in the world, and approximately 0.6 million deaths have occurred till date due to the diabetic condition. And it also puts the high burden on healthcare system in India due to these diabetic conditions. So there are major factors which are affecting to the diabetes development of the diabetes, like inflammation, altered lipid metabolism, oxidative stress, which is resulting due to the wrong dietary habits and sedentary lifestyle. Recently, the gut microbiome has been identified as a major contributor in the development of diabetic and its complications. Previously, we have published this data where we found that the abundance of the, in the observed diversity, alpha diversity, we found that the alpha diversity was decreased significantly in the newly diagnosed diabetic individuals. However, after the anti-diabetic treatment, the diversity is slightly increased and restored to the normal one. And based on the beta diversity analysis, where we found that the inter-individual variation is more in diabetic individuals compared to the pre-diabetic and healthy one. However, pre-diabetic is slightly overlapping with the diabetic individuals. So based on this, we have proposed the study where it is known that previously is known that oxidative stress is involved in the development of T2D. Among various parameters of oxidative stress examined previously from our lab, we found that the glutathione was the best covariate for the glucose recovery. It is also published in one of the journal where we have conducted a trial for around 250 individuals where we found that the GSS lowers the glycemic condition in the type 2 diabetic individuals. So here in this study, there are a control as well. This is a case control study where control individuals, which are known as diabetic alpha for the day 0, and control individuals after 6 months are known as diabetic gamma. Then case individuals were like diabetic plus they have given the GSS supplementation of around 500 mg per day, were termed as the diabetic glutathione alpha. And the group after 6 months is known as diabetic glutathione gamma, like DG gamma. So stool and blood samples were collected. Blood was collected by trained phlebotomists from the Golwilkar Diagnostics Lab in the Pune, and stool sample was collected for the microbiome analysis, where we have for serum biomarkers, we have analyzed the fasting glucose, HbA1c, fasting insulin, GSH and GSSHG, and 8-OHDG, which is oxidative DNA damage marker. And from stool sample, we have extracted the microbial DNA and went for the 6S RRN gene for the next generation sequencing using Illumina platform, and analyzed using the Data2 platform for the microbiome profiling. In serum biomarkers, we found that after glutathione supplementation, in DG gamma group, after 6 months, the fasting glucose and HbA1c were significantly decreased after long-term supplementation. However, the GSH was increased significantly after 6 months. It suggests that the body stores of GSH was maintained in the DG gamma group after 6 months of treatment, where 8-OHDG was decreased significantly after the GSS supplementation. It suggests that the DNA damage marker is decreased drastically after the treatment, leading to recover the oxidative stress in the body, reduce the oxidative stress in the body. However, HOMA beta was also increased in treatment group compared to the control. It suggests that the beta cell function of the individuals were recovered compared to the normal diabetic individuals. Then we moved to the to analyze the microbiome of the these individuals, and we found that the phylogenetic diversity was not changed significantly in both the groups. Then we moved to check the Simpson dominance for the dominant taxa which are present in the overall gut microbiome, and we found that the dominance taxa were increased significantly in DG gamma group after the oral supplementation. However, the inter-individual variation in the beta diversity was seen in this group. You can see that. Then we moved to analyze the overall microbiome profile at the phylum level, and found that among the top phyla, we found that Proteobacteria was found to be significantly decreased after the treatment, 6 months treatment. Then the genus level, the general level microbiome analysis was done, and we found that the genus Bacteroides megasphaera was found to be significantly abundant with increasing trend, while genus Escherichia and shigella exhibited the decreasing trend in subjects with the glutathione supplementation. So previously we found that the body stores of glutathione was increased in the individuals after treatment. Then we have decided to check the effect of this glutathione supplementation at the intra-individual level, and we found that in DG group, where we are given the supplementation, the decrease in the Escherichia ASVs, that amplicon sequence variant, was found to be decreases in each individuals, around almost all the individuals in DG group. And however, the beneficial microbes like ASV2 for the megasphaera were found to be increasing in all the diabetic individuals after the 6 months treatment. So the final conclusion is, this is the first report demonstrating the shift in the gut microbiome community structure and function from healthy to diabetic state in the Indian population. Our findings suggest that the gut microbiome diversity significantly decreased in new DMs, but not in pre-DMs, and was restored in non-DMs on anti-diabetic treatment, or to some extent. The glutathione supplementation given to each diabetic individual, which lead to increase in the body stores of glutathione and improvement in glycemic parameters, also help in enriching the beneficial gut microbiome and the depletion of the pathogenic microbes. However, this GSS supplementation can be used as an adjunct therapy for the individuals with T2D to restore the healthy gut microbiome. I thank my all the study participants and both the mentors, Professor Saroj Gaskarbi from the Savitribai Phule Pune University and Professor Yogesh Sauche, for their support and guidance to the entire study. Thank you.
Thank you, Akshay. We now move to the last presentation for the third prize, and this will be by Aditi Goel from the Central Food Technological Research Institute, CFTRI. Aditi, please come. Thank you.
Good afternoon, everyone. So today I'm here to present my poster entitled Vitamin A Enriched Probiotic Carrot Nectar Regulates Gut Microbiota and Reduces Obesity, a leading cause of cardiovascular diseases. So obesity is a global health issue in the developed and in developing countries. And according to World Health Organization, presently there are 600 million obese adults, which almost represents 13% of the world population. And this number is expected to rise over 1 billion by year 2030. Obesity is considered as a root cause for many non-communicable diseases like cardiovascular diseases, and NAFLD, type 2 diabetes, and arthritis. So it is very important to detect and prevent the obesity at very early level using various markers like increased BMI and dyslipidemia. This will help the prevention of many metabolic disorders. Though FDA has approved many anti-obesity drugs, but they are associated with some side effects in the long term. And probiotics is widely studied for the anti-obesity effects. And combining these probiotics with the suitable food metrics can help in the prevention of obesity. According to a global healthcare company, a weight reduction of 5% will lead to prevention of hypertension and hyperglycemia. And if a person is able to manage the weight reduction of 10 to 15%, a positive effect can be observed in the cardiovascular diseases also. So to develop a natural anti-obesity therapy, we have screened several fermented food products collected from the Sikkim and isolated many probiotic bacteria. And finally, we have selected one with a good cholesterol reduction potential. This Lactobacillus plantarum MCC5231 was earlier characterized for the production of bacteriocin, which was active against a range of foodborne pathogens and multi-drug resistant bacteria. It is shown to have good sustainability in gastrointestinal tract based on its acidic and bile tolerance and binding ability to the mucin. And it is also characterized under in vitro conditions for the cholesterol reduction potential. And based on the European Food Safety Authority's guideline, if a probiotic bacteria is to be used for the consumption purpose, it has to be analyzed for the safety assessment, and after the following the guidelines of authorities, it is found to be safe for the consumption. So this is the probiotic carrot nectar, which is developed after the incorporation of probiotic bacteria into the carrot nectar. And this probiotic carrot nectar can be stored for 75 days under refrigerated conditions with a viability of more than 6 log CFU per ml. So to see the anti-obesity effect of this probiotic carrot nectar, C57BL6 mice were used, and they were fed with high-fat diet for first 8 weeks to make them obese, followed by the probiotic treatment for another 8 weeks. So the mice were divided into the 6 groups. Only first group was given the normal diet, rest all the groups were given high-fat diet. Once the mice gained appropriate weight, the probiotic treatment was started. In group 6, the probiotic product was fed, and in group 5, only probiotic bacteria was fed. Whereas in group 4, only carrot nectar without the incorporation of probiotic bacteria was fed to do a comparative study. Whereas in group 3, a reference bacteria, which is already studied for such kind of benefits, was used. Now, the measurement of physical parameters is first visual appearance, which tells about the effectiveness of the probiotic bacteria. So weekly body weight was taken during during anti-obesity, during the studies. And we found that the high-fi diet has increased the weight from 25 to 46, which is reduced to 82% on giving the treatment directly through the cells. Whereas this reduction in weight was even higher when the probiotic product, which was incorporated with the cells, was given. The similar effect can be seen in the reduction in organ weight, especially in the adipose tissue, where the weight reduction of weight was reduced to 77 to 75%. And similarly, abdominal circumference was reduced to almost 85%. Now, as we know that the gut microbiota is a major reason behind several such diseases, so to see the changes in the gut microbiota, the weekly fecal sample was collected and was analyzed for the changes in gut microbiota. So feeding the mice with high-fi diet resulted in gut dysbiosis, whereas when probiotic treatment was given, the lactic acid bacteria count was increased, whereas the harmful pathogens like Staphylococcus aureus and E. coli was reduced. Now, our aim was to count the number of Lactobacillus plantarum present, which can be a contributing factor for this. So we have done the quantitative PCR using Lactobacillus plantarum specific primers, and we found that high-fi diet was containing 4 log CFU per ml of Lactobacillus plantarum, whereas the probiotic treated group were containing 8 log CFU per ml of probiotic bacteria. Then the changes in serum lipid profile was measured. So the treatment with probiotic bacteria has reduced the total cholesterol levels to 81.3%, whereas in probiotic product fed group, the level was reduced to 75%. And similar changes can be observed in the levels of triglyceride and LDL, whereas the amount of HDL, which is considered as a good lipid, has increased. SGOT and SGPT are the markers of hepatic dysfunction. When there is necrosis in the liver, these markers are leaks out into the blood, which results in increased serum level of these markers. And this is what we have observed in our studies. In high-fi diet, the level has increased in comparison to normal diet, whereas on giving the probiotic treatment, the serum level of these markers were reduced. Now, as we know that the gut dysbiosis will lead to the oxidative stress and inflammatory conditions. So to see the immune response of the mice during high-fi diet, the inflammatory markers were checked. And what we have observed is during high-fi diet feeding, the inflammatory markers like IL-6, IL-12, and TNF-alpha were increased, whereas the amount of IL-10 was reduced, which is reversed on giving the probiotic treatment. And most of the probiotic bacteria have good application potential in reducing the oxidative stress by regulating NRF-ARE2 pathway. And so giving the probiotic treatment to the obese mice has reduced the oxidative stress conditions in the liver. The oil red staining for the liver tissue was done to see the deposition of lipids. And in high-fi diet, the red color droplets represents the deposition of fat. As per the scoring, the 67% of the liver of high-fi diet group mice was accumulated with the lipids, whereas the probiotic treatment has reduced this accumulation to 37%. The similar findings were observed in H&E staining, where the second figure is the liver, and in HFD group, we can see the vacuolar structures, which represents the deposition of fats. The similar structures can be observed in NPP group, which was fed only with the carrot nectar without addition of probiotic bacteria. And these vacuolar structures were reduced in LP group, whereas when the treatment was given through probiotic product, these vacuolar structures were completely removed. Now to find out the mechanism how this probiotic bacteria will be working on the liver and adipose tissue, several obesity associated markers were checked. And finally, we concluded that when there is gut dysbiosis, inflammation, and oxidative stress in the body, it will lead to the disbalance of leptin and adiponectin into the adipose tissue. And this disbalance of adipokines will result in disruption of metabolism in adipocytes, that is AMPK pathway, which was regulating the lipid homeostasis in the liver through PCSK9 and SREBP2. And therefore, now the liver is not able to metabolize the lipids properly, and it will be accumulating into the blood, leading to increased levels of fat in the body. And against this, the probiotic bacteria is working. So conclusion is probiotic carrot nectar is a symbiotic beverage where probiotic bacteria and fiber-rich carrot act symbiotically to regulate lipid metabolism and fat accumulation via modulation of gut microbiota and immunomodulation. Thank you.